All right, we're going to go ahead and get started. We have a very full schedule here, and first of all, I'm sorry that we're going to probably run out of seats, but I hope folks will stick around. So there's some room up front if you want to come up here towards the front. So my name is Gary Wu. I'm from the University of Pennsylvania, and so welcome to our research caucus briefing on overcoming drug-resistant bacteria, fecal microbiota transplantation. A couple of quick introductions. So we're going to have 3 talks. Each one's going to last about 15 minutes. And what we'd like to do, because the timeline is pretty tight here, is we want to hold questions for a panel discussion for about 10 or 15 minutes after all 3 talks. So all 3 of us will come up afterwards, and you can ask your questions, and we'll do our best to answer them. So there are going to be 3 talks. I'm going to give the talk on the gut microbiome good bug, bad bug story. Ed Lautenbach from the University of Pennsylvania will give a talk on antibiotics and development of drug-resistant organisms. And then Colleen Kelly will talk about current and future FMT therapies. So this presentation is being brought to you by the American Gastroenterological Association. So we're the trusted voice of the GI community. It was founded in 1897, and it really represents over 16,000 members worldwide. And our Center for Gut Microbiome Research and Education worked with the AGA to put together this caucus briefing. And our mission is really related to human health and disease and thinking forward about how we can use the microbiome for those purposes. So here's just information about how to get a hold of us. So I'm going to start with my talk here. And what I'm going to do is basically give you an overview of the microbes that live in your gut and why, from a GI perspective, this is actually relevant. So the gut microbiota we think of as largely bacteria, but it turns out that there are many different microbes that live in your intestinal tract. Viruses, fungi, and yeast, and another life form you may not be aware of called archaea. In fact, there are probably more viruses in your intestinal tract than there are bacteria, but these are viruses that infect largely bacteria and not us. Now, the microbiota is enormous in your intestinal tract. It contains about 100 trillion bacterial cells, which makes your intestinal tract probably one of the most densely populated microbial communities on the face of the earth. The genome size of the microbiota is at least 150-fold greater than ours as a human host, which means that it has the genes and the genetic ability to produce a lot of small molecules that we're not able to produce, that we actually end up absorbing, and I'll talk a little bit more about that in a couple of minutes. Now, we have co-evolved with our gut microbiota to live in a mutualistic relationship. These bacteria and these microbes in our intestinal tract are really important for health. We provide a lot of benefits to the bacteria, provide a unique environment in which they live. But in return, bacteria provide many important physiologic processes that are important for human health. So even though we're associating the gut microbiota with various types of diseases, it's actually very fundamentally important to recognize that they are really, really important for health. So this is sort of my 30,000-foot overview of our interaction with the gut microbiome. I'm sure it's a little bit of a complex slide here. You can see that on top, all those little microbes, that's the lumen of your gut. That's what lives in the exterior environment of your intestinal tract. And in the bottom, all those other cells there are the immune system, the cells in your body that respond to those bacteria. And that's the internal environment of the host. And the remarkable thing is what separates us from all the microbes in your gut is largely one cell layer thick, and that's called the intestinal epithelium. Now, our intestinal epithelium produces products and proteins that interact with the microbiota. In return, the microbes actually interact with us. So on the far right over there, you can see that certain types of microbes have an anti-inflammatory effect. They reduce inflammation. Other types of microbes induce more of a pro-inflammatory effect. And on the left side, you can see that microbes can program specific cells in your body to produce certain types of proteins that, again, help to protect us from all these microbes in the environment. Now, the important concept for the rest of my talk is that what you eat in terms of diets, antibiotics, and other things actually not only shape the types of microbes that live in your intestinal tract, but serve as a substrate for these microbes to produce a lot of small molecules, those metabolites that we actually then end up absorbing and circulate throughout our body. So the notion is, together with the ability of the microbes to shape the immune system, the ability of these microbes to produce all these small molecules that we end up absorbing are playing a role in the increasing incidence of various types of diseases that we see worldwide. And this is only a small list. There are many types of diseases associated with living in an industrialized nation that have been increasing very rapidly over the past several decades. So the question is, why is that? Well, that's not a genetic effect on us. It takes thousands of years for our genome to actually evolve. This must be a very strong environmental effect. So because a lot of these diseases are associated with inflammation, the notion is that our microbial environment has somehow been changed. And because of that, we now are predisposed to develop some of these immune-mediated diseases. And this is known collectively as sort of the hygiene hypothesis that I show you on this slide here. So on the left side here is the infant that is born largely sterile. So if you're an infant born largely into a normal microbial environment, you develop a normal immune system, normal immune tolerance, and if you get infected, you get inflammation, the infection goes away, and the inflammation goes away, and you're largely healthy. That's what's on the left side of the slide. But on the right side of the slide is an infant that's born into an abnormal microbial world. And our microbial world is now a lot different than it was a century ago. We're living in a much more sanitized environment. We're using many more antibiotics. Our diets have changed. There's a plethora of reasons for why our microbial environment is now different. Because infants now fail to establish a normal microbiome, the hypothesis is that we now develop an inflammation, autoimmunity-prone immune system that somehow later on in life, due to another exposure, we now begin to respond abnormally to microbial products, and we develop these chronic diseases like Crohn's disease, asthma, and many other types of diseases. That's the notion. So one thought is that actually exposure early on in life to our microbial environment may predispose us to development of diseases later on in life. Well, this is brought to you by the AGA. So we think about gut type of diseases, for example, and inflammatory bowel disease is one of those diseases that affects about 1.5 million people in the United States alone. It's largely divided into two types of diseases: ulcerative colitis that affects only the colon, and Crohn's disease that is inflammation that involves any region of the intestinal tract. These are debilitating chronic inflammatory diseases that require lifelong immunosuppression. Now, on the right side, on the right side is the pathogenesis. What causes inflammatory bowel disease? Well, like a lot of diseases, it's very complex. There's a genetic predisposition that leads to immunoregulatory defect in response to an environmental trigger. And we think the gut microbiota, the microbes in the gut, are the most important environmental factor. So all our current modalities for treatment of inflammatory bowel disease are deep immunosuppression that affect us. Yet we're not really focused on changing the environment of the gut for a treatment modality in inflammatory bowel disease. Yet we know, and that's what's on the bottom of the slide here, that the genetic contribution to the development of inflammatory bowel disease is a relatively minor component. So for Crohn's disease, it's at most 30 to 40%. And scientists have identified nearly 200 genes that are associated with inflammatory bowel disease. All those genes together only account for about 13% of the risk for development of Crohn's disease. So what does that mean? That means the environment of the gut, diet, the microbes in your gut probably are the biggest factor in the development of inflammatory bowel disease, yet our current therapeutic modalities are not focused on engineering the environment of the gut. Just to give you an idea of how important this environmental effect is in Crohn's disease. This is the increasing incidence of Crohn's disease worldwide. So the incidence of Crohn's disease began to rapidly rise in the 1940s, largely in industrialized nations such as the United States and Europe and in Israel. But as the Japanese became more industrialized, several decades later, they begin to see a rapid rise. And now, in China and India, they're on the upward steep slope. And so that, in fact, they're tremendously worried about this increasing incidence of these inflammatory diseases, such as Crohn's disease, in their countries also. So these, again, are very strong environmental effects. So what really drives this inflammation from the gut microbiota standpoint? Well, on the top right-hand corner is the notion of the different types of microbes that can be sort of classified in very broad categories. There are symbionts, which largely have beneficial effects. There are commensal organisms that largely don't bother us. We don't bother them. They sort of live in a mutualistic relationship with us. And then there are pathobionts. These are organisms that are deleterious that can cause disease. So in the bottom left-hand corner, you can see that the colors of the different types of microbes in IBD is actually very different from normal. And so we know that the microbiota is very different in patients with IBD. This is known as dysbiosis, abnormal community structure of the microbes associated with disease. And the notion is that in inflammatory bowel disease, because there is a dysbiosis, that there is an imbalance, and that on the bottom right-hand corner, you can see that there are more pathobionts that are leading to inflammation rather than sort of the healthy beneficial microbiota. So that dysbiosis is in bad imbalance. So if that's really the case, and that some of the inflammation is in inflammatory bowel disease is due to that dysbiosis, our strategy is to change that dysbiosis, to make it healthier to treat chronic inflammation, such as inflammatory bowel disease. So we can think about many different ways of doing this. You're going to be hearing about a number of these things. I'm going to just talk about dietary intervention for a couple of minutes. So our interaction in terms of the nutrients we eat with the gut microbiota is actually exceedingly complex. That's shown on the left. You can't read any of that. That's not important. But there are some standard reproducible findings that scientists have found. One is that there's a decrease in microbiome richness. That's a decrease in the numbers of various types of bacteria and their genes that are associated with both disease states and a consumption of a westernized diet. So what do I mean by that? I mean that individuals with marked obesity, insulin resistance, dyslipidemia, inflammatory phenotypes have low bacterial richness. They have less complex. They have less different types of organisms. Increased consumption of agrarian diet rich in fruits and vegetables, high in fiber, have an increase in bacterial richness. And energy-restricted diets increase bacterial gene richness. And this is essentially what we see in agrarian cultures such as Africa, which eat primarily a plant-based diet. Those individuals have a richer microbiota and have a lower incidence of many different types of diseases that are associated with industrialized nations. So from the standpoint of inflammatory bowel disease, there is a diet that we can use to treat Crohn's disease. This is used as first-line therapy in Europe, only a couple places in the United States. These are elemental diets, largely cannot be consumed orally. They're fed through a tube into the stomach of the patient. And these are basically pre-digested diets. They work actually remarkably well in Crohn's disease, but we don't understand how they actually work. So in the pyramid on the top, I show you our current paradigm for trying to understand how diet through the microbiota may be beneficial in inflammatory bowel disease. So we know that diet is associated with inflammatory bowel disease. And we know that gut microbiota plays a critical role in the development of inflammatory bowel disease. And I just showed you a couple of slides linking diet to the gut microbiota. A lot of scientists are thinking very hard about how we can study current diets that work in inflammatory bowel disease to really understand proof of concept. What is it that they're actually doing? And because if we could understand those principles, we could define better diets for patients with inflammatory bowel disease and other diseases. So one notion is that these defined formula diets for the treatment of Crohn's disease are most effective when they're consumed completely in place of a normal diet. That's why we call it exclusive enteronutrition. So you can't eat any whole food. You just use that liquid diet. But that brings up two very fundamental questions. Does that liquid diet provide something good for patients with IBD that's not abundant in regular food? Or does the consumption of that diet, that liquid diet, exclude something that is bad in patients with IBD that's in our regular diet? Maybe there's something in our diet that has been introduced over the past several decades that's actually bad for patients with inflammatory bowel disease. And there is growing evidence now, only in animal models, that certain things have been introduced into our food supply. And here I give you examples of dietary sweeteners or emulsifiers. Emulsifiers are those things that give ice cream its creamy taste, or salad dressing. It keeps them sort of mixed together. It's almost ubiquitous in any processed food that you eat. There's evidence, at least in animal models, that these things change the microbiota and lead to inflammation. Now, I'm not saying that artificial sweeteners and dietary emulsifiers cause inflammatory bowel disease. All I'm saying is that our food supply is a lot different now than it was a century ago, and some of these things may have unknown effects on us. So our notion is that diet can directly affect the host. Diet can affect the microbiota. Diet, the composition, how long you've been on a diet, not only affects the different types of microbes that live in the environment of the gut, but also help to produce small molecules that we absorb that may affect our physiology. And then thinking forward, we can think about medical foods that may directly affect us as the host. We can think about the next generation of prebiotics that may change the composition of microbiota or serve as the substrate for the microbes to produce those small molecules. We could think about the next generation of probiotics deeply changing the composition of the microbiota for health, or a combination of both prebiotics and probiotics together. So with that, we're going to move on to our second talk by Dr. Ebbing Lautenbach, who's chief of the Infectious Disease Division at University of Pennsylvania.
Great, thank you, Gary, and good morning, everybody. I suppose good afternoon now. So there's a reason why I, as an infectious diseases person, is flanked by two gastroenterologists on either side of me, and the reason for that is that while Gary's done a really nice job of defining a lot of the clinical issues that relate to the microbiome and the gut, inflammatory bowel disease, for example, we, from the infectious disease side, are really interested in what goes on at the level of the gut as well, for reasons that I'll talk about in a second. But if you think about most of the infections that typically will affect all of us, whether it's GI infections, whether it's urinary tract infections, where most of the bacteria come from that end up causing those infections are from the gut. And the effect that antibiotics have on the emergence of antibiotic resistance, which I'm going to spend the bulk of my time talking about, that really happens at the level of the gut in any given person. So that's why the gut, in particular the microbiome, is really interesting to ID folks as well. So this is a schematic of the timeline of antibiotic development. So as you know, the antibiotic era is still not even 100 years old, and so the things that used to kill people with a good amount of regularity, once antibiotics were introduced, that in many ways represented one of the biggest advancements ever in medicine. So with the introduction of the sulfonamides, aminoglycosides, penicillins, cephalosporins, things that all of us and our families use periodically, there was a large amount of antibiotic development over the course of several decades. On the top of the schematic is the development of antibiotics and sort of when in the time scheme those occurred. What you see on the bottom is the emergence of resistance to those antibiotics. And what you can tell pretty quickly is that it didn't take long for any antibiotic to get introduced into the market before we started to see bacteria that were resistant to those antibiotics. And so in many respects, as smart as we are about coming up with novel antibiotics and antibiotics that work in new ways, it actually didn't take bacteria too long to figure out how to get around those things. What you also see from this timeline, which I'll come back to in a second, is that while there's a rash of development of new antibiotics over the course of the first 50 years or so, there's been a marked slowing the development of new antibiotics. And so if one of the approaches for addressing antibiotic resistance is simply to develop new antibiotics, that's a strategy that's not going to work in the long term, because most of the easy compounds, the easy antibiotics to develop because they're based on things that already occurred naturally in the environment, all of those have been developed. And so the strategies for developing new antibiotics are really complex. So there are a lot of different mechanisms by which antibiotics work. They may work on disrupting the cell membrane of bacteria. They may work on affecting protein synthesis of the bacteria. They may work on how the bacteria replicate. So there are a lot of different ways in which antibiotics work, and different classes of antibiotics work in different ways. As I mentioned, it doesn't take long for bacteria to figure out how to get around those mechanisms of action. And so bacteria have been really ingenious about developing novel mechanisms of resistance. So whether they affect the antibiotic's ability to even get into the bacteria, many bacteria work by that mechanism. If an antibiotic is able to get into the bacterium, there are mechanisms that bacteria have developed to actually pump antibiotics out of the bacterial cell. So there are a lot of ways in which bacteria have figured out ways around the kind of antibiotics that we've developed. If you look at the emergence of antibiotic resistance, these are data from the CDC, which are now a couple of years old, but these are nationally representative data from a variety of hospitals all across the country. And what they do periodically is to track trends in the emergence of different types of resistant bacteria. So whether it's methicillin-resistant staph or MRSA, vancomycin-resistant Enterococcus, or multi-drug-resistant gram-negative organisms like Pseudomonas or Acinetobacter, for almost every bug-drug combination, there's been a significant increase over the course of the last 20 years. Some more than others, but almost without exception, there have been marked increases in these. Now, these are mostly hospitalized patients. That's where these data come from. If you look at community isolates, so E. coli, which most commonly causes urinary tract infections, MRSA is obviously a big problem in the community as well, marked increases in those resistant organisms as well. It's not limited to the United States. These are data from Europe, which show very similar trends, although the Europeans tend to be a little bit better in terms of using antibiotics a bit more judiciously than we do, which I'll come back to in a second. Even with that, marked increases in all kinds of different bacteria. These are data from the ICU, but if you look at European data from hospital settings outside of the ICU and even community settings, there have been significant increases in resistant organisms. When you look at the United States, this is Klebsiella pneumonia. Klebsiella pneumonia tends to cause urinary tract infections, can cause pneumonia, causes a variety of different types of infections. And what I'm showing you here, and these are again data from the CDC, is how these break down geographically within the United States. And also broken down by whether the organism is resistant to 3 classes of antibiotics or 4 classes of antibiotics. By the time an organism is resistant to 4 classes of antibiotics, by and large, that's a bug that we have a really hard time treating, for sure in the community, but even in the hospital. So there are an increasing number of these organisms that when we're called to go see them in the hospital, we don't have a great choice of an antibiotic with which to treat people. So we end up picking it's sort of a kitchen sink type of approach where you end up taking 2 or 3 antibiotics, none of which by themselves work terribly well, but you hope that in combination they're going to treat the infection that you've got. So these are really big problems. The reason for the geographic variability is somewhat unclear, although probably relates somewhat to differences in how antibiotics are used by geography. And in fact, there are differences when you look at the Northeast versus the South versus the West for how people use antibiotics. And there are also some differences in transmission patterns, so person-to-person spread of different organisms. Similar data here for Pseudomonas aeruginosa and Acinetobacter baumannii. These are both, like Klebsiella, gram-negative bacteria, which also have increasing amounts of resistance both to 3 classes and to 4 classes of antibiotics. And again, some geographic variability for the same reasons as Klebsiella. If you look at the overall proportion of resistance among Klebsiella, Pseudomonas, and Acinetobacter, again, marked increases in resistance over time, and this is prevalence data now from a couple of years ago, demonstrating that increasingly we have organisms that we have a very hard time treating in the hospital, for which there are oftentimes no antibiotics available to treat. one of the things that that's become increasingly evident is that although I've defined things by what's going on in the United States or what's going on in Europe, this is really a global problem. And when you look at organisms that are identified in various parts of the globe, what you see is a lot of similarity in the strains that we see across these disparate geographic regions. And the reasons for that is really increased globalization. So globalization of obviously humans moving back and forth, but products moving back and forth as well. So this isn't a problem that is U.S. specific, but really stretches every corner of the world. As I mentioned, one of the strategies that we use to address resistance, or at least have historically, is to encourage the development of new antibiotics. This is now about a 30-year chart demonstrating the development of new antibiotics. So what you see is when you start off in the mid-'80s, this is the numbers of new agents developed in that 5-year time stretch, significantly reduced over time. And the reasons for that are several-fold and have to do both with financial implications for development of antibiotics versus development of other agents, but also because, as I mentioned, the ability to identify easy new targets in antibiotic development is really negligible. So it's very difficult to bring these drugs to market. And for a variety of financial reasons, these are not the agents that typically are currently emphasized. And so new antibiotic development as a strategy to deal with resistance is really not going to be where it's at for sure over the course of the next 10 to 20 years. So I think the upshot here is that while I mentioned how bleak things were before the introduction of antibiotics, we have a real risk of getting back to a pre-antibiotic era where we have infections that we simply can't treat. We're already there with multi-drug resistant infections in the hospital. But as we see this more and more, and as we run out of available antibiotic strategies, this is a huge problem. And I think we do risk getting to the point where we have an increasingly large number of patients for whom we have infections that we have no strategies. So what are the things that are driving resistance? And this is where I'll start bringing it back to the gut and to the microbiome. So the biggest driver, and these are data based on a variety of consensus panels. There are obviously a lot of things that can drive antibiotic resistance. The biggest driver is antibiotic use. So if you're in the lab and you have a fully susceptible bacterium and you're trying to figure out how to get that bacterium resistant to an antibiotic, start exposing it to small doses of antibiotic and increasingly larger doses of the same antibiotic, that bacteria is going to get resistant, and it'll do it fairly quickly. So both the lab evidence and the clinical evidence for how you get antibiotic exposure and how that drives emergence of resistance is pretty clear. So what are the reasons why we use antibiotics so much? Reasonable evidence both from the inpatient setting and from the outpatient setting suggests that in at least 50% of cases where somebody's using an antibiotic, it's not needed. So there's clearly a lot of room for improvement. That number is probably even bigger in the outpatient setting, where there's still a lot of antibiotic prescribing for what is almost certainly a viral upper respiratory infection, probably something that many of us have right now. Those are overwhelmingly due to viruses, for which bacteria do absolutely no good. And yet, we often prescribe, the collective, we often prescribe antibiotics for that. I try not to. So why do we use antibiotics so much? Well, there are a lot of reasons for it, which I think are pretty evident. So some are patient concerns. So a patient comes in to see you and they have a bad case of the sniffles and they want to know why and they want to know what you're going to do about it. And they have real concerns. So they have family that they have to take care of. They have a job that they want to go back to. And historically, they're like, well, my old doctor always gave me an antibiotic and my symptoms got better after a day or two, so why aren't you giving me an antibiotic? Now, the truth is their symptoms probably would have gotten better after a day or two anyway, even without the antibiotic. But it takes time to explain that. So what are the physician concerns? In general, physicians assume that patients want antibiotics. Increasingly, there's some reasonable evidence that physicians probably overestimate that as an incentive for patients. I think what patients really want is an explanation. They want you to talk to them as a physician and say either, I think you need an antibiotic and here's why, or I don't think you need an antibiotic and here's why. The diagnostics that we have to tell you whether you have an infection and whether it's bacterial or not are pretty much the same diagnostics by and large that we had 30 years ago. So diagnostics is a real problem in infectious diseases, and that's why antibiotics are used a lot because if we don't know for sure whether somebody's got a viral or a bacterial infection, chances are we will hedge on the side of safety and say, well, you know what, I'm probably not going to do much bad by just giving them a short course of antibiotics, and even though I think it's a virus, but I'm not sure. And then there's time pressure. So as patients are expected to get through being seen by a physician quicker and quicker, it's a lot easier to write a prescription for an antibiotic. That takes half a minute than to spend 10 minutes explaining to the patient why they don't need an antibiotic. As I mentioned, antibiotic use leads to resistance. So the more antibiotic exposure that you have as a given patient, the greater your likelihood of either having your own bacteria start to develop new to develop resistance mechanisms, or it makes that patient more likely to have new bacteria acquired from other patients, especially if you're in the hospital. The people who are at highest risk of this are the most immunocompromised, so people who are on chemotherapy, who are on other immunomodulators because they got a transplant, or if you have an indwelling device, so a urinary catheter, a central venous catheter, also places you at much greater risk. This is sort of what I talked about before. This is basically the selection of antibiotic resistance. So you expose either in the lab or in the clinical setting, you expose somebody to increasing amounts of antibiotics. Their bacteria, and these are typically the bacteria that are colonizing the GI tract, are more likely to start becoming more resistant. So remember that even if you're using an antibiotic for the right reason, so somebody's clearly got a urinary tract infection and you give them an antibiotic, which is what you want to do, you want to treat them, that antibiotic isn't just going to the urinary tract. It's not just going to treat that bug that's in the urine. It's going throughout the body. So it's going to the lung, it's going to the bloodstream, it's also going to the gastrointestinal tract, especially if you have an antibiotic that you're taking orally. So all of the bacteria that are in your body, not just the ones that are causing the urinary tract infection, but all of the bacteria, the trillions and trillions of bacteria that Gary talked about in the gut, are also seeing that antibiotic. So what happens? The bacteria that are susceptible to that antibiotic are going to get wiped out. So even that simple antibiotic that you give for a urinary tract infection is going to wipe out a lot of the bacteria that are in the gut. So what happens? The other bacteria that are sitting in your gut that are already resistant because they have a natural mechanism of resistance already in place, they're the ones that are going to stick around. So what are the bacteria that are now going to colonize your gut? It's fewer of the healthy bacteria and more of the bacteria that are already resistant to antibiotics. What happens if you get another course of antibiotics after that with a different antibiotic? The same thing, except that now you're selecting for a more and more resistant population of bacteria, and that's what happens. So why is it really important to focus at the gut? For all the reasons that I started off talking about, which is when we think about where the bacteria come from that cause infections, it's almost certainly at the level of the gut. So while we may know that you have a urinary tract infection with an E. coli that's resistant to X antibiotic, we don't know what's going on in the gut because we don't typically look for that. And so there really is sort of an iceberg phenomenon in that while we may know that you're colonized in your urine with a particular organism, we don't know what's going on at the level of the gut unless we go looking for it. The same way in which you can elicit more resistant bacteria in the lab, think of what goes on at the gut as basically the lab in a clinical setting. So that's why it's really important to look at colonization. And the other reason that it's important is by the time the horse is out of the barn and you've given somebody an antibiotic and now they're colonized with a resistant bug, one might assume that, well, when I take that antibiotic away, they're probably just going to get colonized with all the healthy bacteria again. Not necessarily. Once you're colonized with a resistant bug, and although there aren't a lot of studies that have looked at that, the duration that you're colonized with that resistant organism is on the order of months to years, not days. And even if you can't find that resistant bug in your gastrointestinal tract anymore, when you get another course of antibiotics, oftentimes that bug pops right back up again. And so this is really a message of try to prevent that colonization from ever happening, or if it happens, then think about novel strategies for getting rid of that colonization. I think that's why studying the gut microbiome and figuring out what the healthy microbiome is as it relates to antibiotics and antibiotic resistance is so critical. And I think that is that's actually what I said in this last slide. So let me stop there.
Thanks, Eb. And so our next speaker is Colleen Kelly, and she's going to tell us about fecal microbiota transplantations, very much related to what Eb just talked about.
Thank you, and good afternoon. I have one of those viral infections. I woke up without a voice this morning, but came back, fortunately. And I'm going to talk about C. difficile infection, and it's really the most basic example of what can happen when your gut microbiota get disrupted by antibiotics. And fecal microbiota transplantation, or FMT, which has emerged as a really exciting and effective alternative to antibiotics for this infection. So a little bit on the scope of the problem. C. diff is now the most common cause of hospital care-associated infections in U.S. hospitals. It's outpaced MRSA for that distinction, responsible for at least $4.8 billion in excess healthcare costs annually. And in 2011, it's estimated that there were 453,000 cases and 29,000 deaths in the U.S. annually related to C. difficile infection. Two-thirds of cases are healthcare associated, and the highest incidence is in women, persons over 65 years old, and whites, and that may be related to differences in healthcare access. And there was a staggering rise in C. diff between 2001 and 2010, where cases of hospitalized patients for C. diff doubled, and deaths also paralleling that doubled as well. And really scary is we're seeing this in populations that were previously thought to be at low risk. So now we're seeing C. diff occur in, a third of cases in patients who've been out in the community without much healthcare contact. When I was graduating medical school in 1999, it was patients like this little old lady in the hospital that you had to worry about getting C. diff. But now I'm seeing it in women who may have had one dose of antibiotics around a C-section when they went in to have a baby. I've had college students who had some dental work done and got C. diff. And even in children, my pediatric colleagues are telling me, that it's on the rise in that group as well. And that was a group that was really previously thought to be pretty protected and didn't get C. diff that often. So C. difficile is a bacteria, and it occurs as a part of our gut flora in about 3 to 5% of people. You may have C. diff inside you, and it's generally kept in check by that diverse community of microorganisms around it. But it's when there's exposure to antibiotics that alters those good flora, kills off those good bacteria, that allows C. diff to just grow crazy, proliferate, begins to produce a lot of toxin. That toxin causes damage to the wall of the colon that results in severe diarrhea. Patients can end up dehydrated, hospitalized. The sporulation is the way the bacteria multiplies, and they basically allows for a very rapid spread of C. diff through hospital settings. And it's funny, it's a disease caused by antibiotics, and yet we've, for 60 years, treated it with antibiotics, particularly antibiotics that have activity against C. diff. And on the cheaper end, metronidazole or Flagyl for a couple of dollars a day, but then vancomycin and more recently fidaxomicin that are used in more severe or persistent infections. Fidaxomicin can be $2,000 to $3,000 for a 10-day course. And there are definitely challenging cases that we're seeing, recurrent disease, where patients respond to that initial dose of antibiotics but relapse within days to weeks of the antibiotic being stopped. That happens 15 to 35% of the time after an initial infection, and once you get in that cycle of recurrence, it can be very hard to get out of. I've certainly seen patients that have gone on for years suffering from recurrent C. difficile infections and needing to go on multiple courses of vancomycin. Obviously, with the expense of that drug, a huge economic impact. I've seen patients lose jobs over this. It's affected mental health, socially. You have this diarrheal illness where you can't leave the house without worrying about being incontinent, so it has a huge impact there as well. And then we're seeing severe, complicated infections that are really refractory to treatment, and these are happening in very vulnerable patients, frail, elderly, people with a lot of comorbidities, where the choice is either you die of the C. difficile infection or you go to surgery and have your colon out. And a surgery like that is a big surgery. 30-day perioperative mortality for a colectomy for C. diff is 30 to 80%. So, in the past couple of years, fecal microbiota transplant has emerged as a really effective and exciting alternative treatment to antibiotics. And what it is really what it sounds like. You're taking stool from a healthy donor, It contains all of those good beneficial bacteria that the recipient is thought to be missing. You transplant it into the recipient, and those bacteria then find a home in the recipient's colon. They grow, they flourish, and they then do the job of crowding out the C. diff. So patients are really excited about this. I'm sure you've all seen things on the news. There's blogs. There's one called The Power of Poop, and it promotes fecal transplant for everything beyond C. diff, even inflammatory bowel disease and other diseases that are associated with alterations in gut bacteria. There's this fellow on YouTube who tells you how to do it yourself, fecal transplant at home. And I actually updated this morning. I looked, it's now had 62,000 views. So home fecal transplants are happening still. There's guidebooks even that you can tell how to do this at home. So the patients are excited, the media's excited. I, as a doctor, was really excited. I've been doing this for 8 years. I've treated over 300 patients with fecal transplant with cure rates over 90%. And there's nothing that's that effective in medicine, over 90%. But the data's catching up, and it's, doctors like me who've been doing this, and researchers, when you think there was a recent systematic review where they put together over 500 cases, and the cure rates are astounding, close to 90% for C. diff infection, even the most recalcitrant cases. And a randomized controlled trial was published in 2013 in the New England Journal of Medicine. It was a Dutch group that compared fecal transplant to a standard course of vancomycin. And that graph on the right, you can see after 1 to 2 fecal transplants, you're approaching over 90% effective compared to 20 to 30% with standard treatments. So how does it work? The mechanisms are pretty complex. This is the way I explain it to patients. Think about your gut microbiota like a healthy lawn, right? And then C. diff is like weeds coming up on your healthy lawn. And you can put all the weed kill around the lawn that you want, and the weeds go away for a little while, but those little seedling spores, they're around, they remain, and you turn your back and the weeds come back up again. Fecal transplant's like sodding a whole new lawn. But then, that's kind of a simplistic example. And we know the mechanisms for how this works are really complex. Think of it more like a rainforest that's been deforested, destroyed. And you need to bring back all of that biodiversity, all of the animals and insects and plants and trees. That's really what fecal transplant's doing in your gut. And that's why it's really amazing. And considering how complex it is and how effective it is, actually doing a fecal transplant is pretty easy and pretty low tech. You need to identify and screen a donor. And typically we choose healthy, low-risk people who have not been exposed to recent antibiotics. We test those people, obviously, for infections, the obvious things, HIV, hepatitis, but other things that can be transmitted through stool or blood. In some way, you collect the fecal material from this donor, prepare it, usually by mixing it in saline or water, and then find a way to administer that material into the patient. And you can do it through either nasogastric or nasointestinal tubes, endoscopies, colonoscopies, or enemas. So despite, that it's so easy to do, so easy to administer, so effective, it's still not widely available. And why is that? What are the challenges that we're still facing getting FMT out there? Number one, questions around safety. We believe it's safe. I've been doing it, and I don't think I've done anything bad to anybody, but there haven't been a lot of really sort of systematic, long-term studies looking at safety. There's the source of donor material. I mean, finding a donor for each individual patient and running around and getting fresh stool in the morning when you need to do a procedure and when you need to do it, that's a lot of work. And regulatory issues. So for people in the room that are interested in the regulatory aspect, that brings up a lot of questions. How is fecal transplant? What really is it? So as far as safety, there's obvious procedural risks. You're doing colonoscopies, you could perforate somebody. There's risks of infection, and there have been some reported infections that have been transmitted. I mean, we're putting a whole lot of germs into somebody. There have been deaths reported, a fatal aspiration actually, where somebody was getting the fecal transplant by the upper route, vomited it, that went into their lung, and it caused a fatal pneumonia. And there have been infections reported following home do-it-yourself FMTs as well, where people maybe aren't doing screenings, using a friend or family member. Then there's the theoretical risks. I mean, Dr. Wu talked to you about all these other diseases that we think may be wrapped up in gut microbiota, from obesity to inflammatory bowel disease. Could we somehow transfer risk factors from one patient to another by doing a fecal transplant? So more than the benefits, are we transplanting along things that we may not understand yet or may not be able to control? So because there's little perspective or long-term safety data, AGA, together with a couple of the other professional organizations, applied for an NIH grant to form a national patient registry. And we aim to follow about 4,000 patients who've gotten FMT for up to 10 years. There'll be a biobank where we maintain their samples before and after, and we're going to be able to collect a lot of data on the real-world efficacy of this, on maybe safety issues that we may not see when we're just treating a couple of hundred patients, in isolation by ourselves. We're going to share this data with FDA, which will help them with some decision-making, as well as any researchers who want to use either those samples or that patient data to study FMT. So we find out about that hopefully within the next few months, whether we're funded. Next, there's the logistical challenges. I can tell you finding an individual donor for each patient, it can be expensive, it can be time-consuming. We're needing to bring these people in, coordinate screening with the day that we plan to do a procedure, trying to figure out what screening and testing is indicated, because there's no real clear consensus out there what you need to do for testing. No matter what instructions or materials you provide to donors, people do all kinds of things that you can imagine that, sometimes we don't have the stool that we need to do the procedure. People can't always produce, Sometimes you get a performance anxiety, you need to bring in a stool for a doctor to do a procedure on your family member. So those are some logistical challenges, and I've suffered through many of them over the past few years. So in an effort to improve that, there's emerged this concept of stool banks. We have blood banks. We don't make you go find a blood donor when you need a blood transfusion. So Open Biome's a nonprofit stool bank up near Boston, and they've kind of centralized that whole process of finding donors, screening them. They send out stool to doctors that they can keep frozen until they need it to treat a patient. And then the commercial side of things, doing almost a very, very similar thing, but putting it through FDA clinical trials. They sought is Rebiotics, and their formula is essentially just frozen human stool, and they've sought orphan drug classification for this, and their product could be available within a pretty short period of time. Then there's the idea of encapsulated products. Do you need to put liquid stool up into people, or can you just put it in a capsule, either fresh or frozen stool, and have them swallow it, make its way down to the colon where you want it to work? And then do you really need to be putting just the whole stool in somebody with all of those hundreds of thousands of bacteria, or can you identify and isolate what are the essential components, what's making it work? Can we just encapsulate those few organisms? It'd arguably be maybe safer, and if as effective, would make things a lot easier. So there are early phase studies underway that this looks like it could be an effective alternative. So these are all coming down the line. And so sort of lastly, regulatory status. The FDA identifies a drug very broadly. It's anything that you use in the diagnosis, cure, mitigation, treatment of disease, or prevention of disease. And they do include biological products in this definition. So by that definition, fecal microbiota is an unapproved drug and a biologic, and considered investigational. And in order to administer an investigational agent to a patient, you need to obtain something called an IND, an investigational drug application. And I did that for a clinical trial that I recently completed on fecal transplant, and it's a lot of work. Enormous time and administrative requirements, reimbursement issues that potentially could limit access to patient care if doctors are required to get INDs just to do an FMT. And they announced this decision in 2013. There was a lot of pushback from providers, from the professional societies, and from patient advocates who said if you really stick to this IND requirement, you're going to really push FMT underground. There's going to be a lot more do it yourself, and it's going to be arguably less safe. So wisely, the FDA came back about 2 months later and said, well, we still think you need an IND. However, we're going to exercise enforcement discretion, which means you can go ahead and administer FMT as long as it's for C. difficile that's not responding to standard therapies. And as long as you provide informed consent to the patient, tell them that it's believed to be an investigational product, and discuss potential risks. So that really opened it up and allowed patients to continue to receive this treatment while the research moves along. So in summary, the epidemiology of C. diff is changing. It's a big problem. It's increasing. It's scary. FMT, though, very effective. Nothing else for C. diff is nearly as effective. But more research is needed, particularly in the long-term safety effects and also potential applications of FMT for other diseases that are associated with alterations in gut bacteria, like inflammatory bowel disease and obesity. And that is all happening. So why are you here? We need your support. Definitely steady, predictable funding increases for NIH. I did have a small NIH grant to do our clinical trial on FMT, and I'm very grateful for that. That allowed research to get done, things to move along. There's this Innovations for Healthier Americans Act, which has already passed the House, and the Senate version is this 21st Century Cures, so encourage people to vote for that. And that's it. So we'll open it up for questions.
Thank you very much, Colleen. Okay, so this is your opportunity for the next 10 minutes or so. Go ahead and raise your hand and
You mentioned diet in your presentation and touched a little on probiotics and prebiotics. Are they really helpful? Some argue that by the time you eat them again, they get to your gut, they're gone.
yes, we were just chatting about that before the symposium. You know, there are differing views on that. My own viewpoint, my personal viewpoint is that when you look at the studies, there are a whole bunch of these studies that are out there. They all sort of trend towards a benefit, but they don't work as well as we want them to work. And the notion is that, well, maybe they don't work as well as we want them to work because largely they pass through, that they're not able to colonize or change things very significantly. So we think that small positive signal is really a great opportunity to think about using the science that we know about the microbiome now to come up with the next generation of products. That's why I emphasize next generation of products that might change things more significantly and lead to greater benefits.
Hi, Catherine Duff. I know you all up there except Eb. We've never met in person. Nice to see you. I'm the founder of the Fecal Transplant Foundation, patient advocate for fecal transplants. When do you think the AGA and the IDSA might get on board with the CDC's recommendations for fecal transplant not being the treatment of last resort? As you know, currently patients usually have to be on their third recurrence or fourth occurrence of C. diff before they're offered a fecal transplant, which results in months and months and months of extended antibiotic use, causing further damage to their microbiota, possibly irreversibly. When do you think that will change and fecal transplant will be offered as a first line of defense?
Colleen, do you want to address that or do you want me to?
I know that the current American College of Gastroenterology guidelines recommend fecal transplant after a failed vancomycin taper. One failed vancomycin taper. And I think part of the hesitancy to recommend it sooner at this point is that lack of the long-term safety data but you could also argue with the long-term safety of taking antibiotics over and over. You know, you could go back and forth there. I think as we move along, as we have more experience with it, as we get potentially safer formulations that are available, I think it may be used earlier in the treatment, in the spectrum there. I've had patients, I guess I take it on a patient-by-patient basis too. So if I have a patient who's had two very severe infections that have landed them in the hospital, the intensive care unit, I usually won't make them have a third episode. I'll make that decision to treat them earlier. And there's nobody that's out there telling doctors you can't use it earlier. This is just sort of suggested. Think about it after they failed this. But there's really no restrictions to using FMT sooner if you think that it's best for the patient.
Most doctors don't understand that, though, out in the general public. They think this is a protocol that's been established and that they have to follow it or they'll get in trouble by the FDA.
And the FDA is definitely not running around chasing down doctors doing FMT for C. diff. There are doctors, and this is kind of another, there are doctors that I know of out there who are doing FMT for things that aren't C. diff in flagrant disregard for what the FDA has said about it, and the FDA is not chasing them down either. And they're charging $10,000, $15,000 cash to patients.
We've heard $78,000.
Yeah, okay, ridiculous amounts of money to desperate people who have illnesses they're willing to fork it over without a lot of data really to support it in these other conditions. So I think that's a big problem. I'd almost rather see the FDA look into those, but I don't think the FDA has the manpower maybe to go off enforcing this.
Thank you.
Can you maybe comment on some of the very, very commonly prescribed antibiotics like tetracycline, used as an acne drug? I've read that comment on that, and also if you could comment on other common things like NSAIDs and birth control pills, which I understand can also interrupt good bacteria. But
Yeah, so they're definitely, I think you can sort of divide the antibiotics that are used into things that are used for defined courses for presumed infection. So a couple of days of an antibiotic for urinary tract infection versus something like acne where you're using, tetracycline, minocycline, or something like that, sort of long term. There are definitely data that not all antibiotics are created equal, so some tend to result in more resistance than others, and some of that is because some antibiotics elicit not only resistance mechanisms that would make the organism resistant to the antibiotic that you're giving, but also elicit resistance mechanisms that cause resistance to a whole bunch of different antibiotics. The data for acne specifically suggests that there are definitely changes in the organisms that colonize people who are on long-term tetracycline, for example. But the environmental impact or the ecologic impact on that patient is less than there is for a lot of other antibiotics. And again, I think in those situations, it's in any given patient, it's a risk-benefit question. So if there's really severe acne and there's good data that giving an antibiotic longer term is going to make a difference, then you weigh that versus the risk. I think the main target for reducing antibiotic use is the use of antibiotics in those situations where it's really not needed. And that's sort of the low-hanging fruit that, even though it's low-hanging, we've had a hard time picking it.
If I could just add very quickly, you brought up xenobiotics, other types of drugs that you might take. Let me just comment on the fact that there's a lot of interest in the gut microbiome and how people respond to different types of drugs, all right? So there's a lot of what we call inter-individual variability in the way people respond to drugs that cannot be explained by us, our own genetic makeup. And one of the thoughts is that part of that is due to the way that the drugs are actually metabolized by the bacteria. So, for example, acetaminophen, Tylenol that you take, there's certain individuals that metabolize Tylenol or acetaminophen in a different way based on the gut microbiota. So that's a sort of a new area of how pharmacodynamics, how drugs are metabolized, in part by the microbes in your intestinal tract. Please, go ahead.
Could you speak to bioaccumulation in drinking water and where you feel like the state of research is and where it should be going? Maybe the whole panel has something to say about that?
Bioaccumulation, you mean chlorination of water and the development of organisms over time that could accumulate?
Bioaccumulation of antibiotics
Oh, yes.
in our drinking water.
Yes, yes.
And
So,
do we feel like we're on the cusp of research, or I'm looking in terms of a request for additional research on it?
yeah, no, I think environmental factors are very critical. So, antibiotics are ubiquitous. I mean, so antibiotics, a lot, as Eb had mentioned, are actually made by bacteria. That's how bacteria fight against each other, plants. It's just the synthetic antibiotics that we've introduced, sort of these heavy-duty, very broad-spectrum antibiotics are now in our environment. And that is a concern, obviously, that it's not only in our drinking water, but our food supply, right? I mean, livestock tend to thrive, grow bigger with low-dose antibiotics, been used for decades, and that's a big concern. And so it's throughout our food supply, and potentially in drinking water, in our environment. And I was going to ask Eb about this, that, we are essentially, in a way, engineering these microbes by treating them with these synthetic antibiotics. And one of the concerns that I was thinking about is that once these are established on planet Earth, some of these organisms have never existed on planet Earth before. Do they ever go away? I mean, they could just be there forever. Like smallpox, you can eradicate smallpox because the carrier is us. But with some of these organisms that live in an environment, they may be there forever, and we just continually accumulate these things on planet Earth. I don't know, Eb, if you have.
Yeah, no, I think that's the exact challenge. I mean, I think even pre-antibiotic era, when you look at bacteria that existed even before we used antibiotics, some of those bacteria had mechanisms that make them resistant to antibiotics, and some of those are functions that serve the bacteria to enhance growth or to enhance adhesion to certain places that allow it to live. So even before antibiotics, bacteria had mechanisms allowing them to become resistant. But I think Gary's exactly right. The sort of organisms that we're creating now, we've demonstrated can live for months, if not years, in people and can do the same in the environment. They're actually pretty hardy. And so I think the challenge is that the more we create these things, we don't have any mechanisms to ultimately eliminate.
We'll take one final question. I think you had a question, and then we'll wrap up.
What recourses are available to patients who are facing the 3 or 4 class resistant infection?
What research, you mean like which antibiotics? So the, just make sure I understand your question.
What recourse? How do you treat them?
Oh, how do we treat them? It really depends on any given patient. And so if there's a definable site of infection, so let's say they have a catheter infection or they have an abscess in their belly, the more that you can get source control of where that infection is, the better. So if you can remove the source of infection, you're generally better. Then there are older antibiotics that we stopped using back in the 50s and 60s. Colistin is a great example. Because it was really toxic, and as we were developing newer antibiotics, we were like, we don't need this antibiotic anymore because it's super toxic. But we're not coming back to using those because in the risk-benefit assessment, if that's the only antibiotic that we have to use, we'll use it. There's some agents that are investigational, and we can get access to those. These are those that companies are developing, but not on the market yet. So on a compassionate use basis, sometimes we can get those. But it really ends up becoming sort of a grab bag for each given patient, trying to figure out what the best combination is. And oftentimes there isn't a best combination. It's whatever you got.
Great. Thank you very much. So we're going to wrap this up. I just want to thank the AGA and our speakers, and especially you, the audience, for great questions. Thank you very much.